There we go. Over to you. Thank you so much.
Okay. All right. Okay. Thanks. So thank you to everyone at Stargardt's Connected and to community for coming and attending this meeting today on behalf of SpliceBio and on behalf of the team. So as a quick introduction, so my name's Jonathon Vine, and so I'm the Senior Director of Clinical Operations at SpliceBio. And so people will say, well, what does that mean? And so very essentially, my job is to set up and run the clinical trials for the Stargardt's development program that we have at SpliceBio as well. So when I say that I manage them, I mean, I must admit that, the other day we actually counted that, I think it's over 150 people that have touched or contributed to even just getting one of these studies set up. So, I'm speaking on behalf of a very, very large team of people and a huge amount of effort that obviously goes into making sure that we can run these studies and obviously, hopefully, have successful studies moving forward as well. So today, the aim is to tell you a little bit more about SpliceBio, for those of you that aren't aware of what we're doing as a company. The gene therapy, and the way that we're aiming to treat the Stargardts, and a little bit more about our clinical trials as well, and the active clinical trials, and potentially, should you be a suitable candidate, whether there's an opportunity for you to join our clinical trials that we have at SpliceBio moving forward as well. Okay. So, I'm based in the UK, although near London, and most of the clinical team are also based in the UK. But of course, SpliceBio is a Spanish company that's based in Barcelona, and so actually, it's on the right-hand side here. You can kind of see part of the Barcelona Research Scientific Park, and we have a very small amount of offices that are in there. Only holds like 10 or 15 people within there. We're a fairly small company, so we have around 35 to 40 employees, although now we are increasing the number of clinical trials that we have, both in Stargardt's and potentially moving forward into other inherited retinal disease programs. We are a growing company. We've been established for around 5 years now, and the technology, the gene therapy technology we're using comes from Princeton University in the US, from the Muir Lab. And so a very famous scientist called Tom Muir, who is one of our founding members as well. And a couple of years ago, we raised an initial amount of 50 million euros, which is enabled us to move forward into initiating our first clinical trials within the Stargardt's development program as well. So, in summary, a little bit more about SpliceBio as a gene therapy company and our technology. So, we are looking to target diseases which are caused by large gene mutations. And so, of course, Stargardt's is a classic example of this. Of a disease that's caused by mutations within a very, very large gene. And the mode of that, the type of gene therapy that we're going to be using is called an AAV dual vector, or adeno-associated viral vector, and protein transplicing. And I'll explain a little bit more detail what that means in subsequent slides. As I said, Stargardt's disease is our front-running program. We do have other inherited retinal diseases that we're looking at that at this point aren't public, but we will be able to provide more information on those in the short to medium term. And I won't go into too much detail today around all of kind of the preclinical data that we've generated within the Stargardt's program at the moment. But effectively, the strong preclinical data that we have underpins our ability to kind of move forward into first in human studies for the first time. And since the last meeting that we've had with Stargardt's Connected, the great news is that we've actually got endorsement to move forward, and we've got the green light to move forward by the regulators in the US, so the FDA, and the MHRA in the UK to move forward into our first in human clinical trials now. And so we have both observational studies and first in human studies, which are now recruiting patients in the US, the UK, and hopefully in Europe soon as well. So I know that a majority of you are probably familiar with how this type of AAV gene therapy works, but for those of you that are new to this, on the slide here, I'm using the analogy of kind of writing a letter and delivering a letter or or information to a reader. And so effectively what we're trying to do when we're writing this letter, or we are creating a healthy version of a faulty gene. So in this case, the ABCA4 gene. And when you have a letter, for example, or you have your healthy gene, the aim, of course, is to deliver that into the human and into the cells. So ultimately, it can produce a healthy versions of the protein that's needed for the ABCA4 protein, which is required. And so, of course, with a letter, the way that you would deliver that is by putting it in an envelope and packaging that with a little stamp and licking the back of the envelope. And effectively, that is what the AAV vector does, the adeno-associated viral vector, is it's a way of packaging that healthy gene so it's ready to be delivered into the human. And so what we do is when we put it in the envelope and we package it, in addition to the healthy gene, we also have to put a few bits and bobs into the envelope as well to ensure that once it's delivered into the human cells, that it stays there, for example, and it's able to express the healthy version of the protein. And so, again, using the analogy of the letter, the next piece is the delivery truck. So, the method that we actually use to deliver the gene therapy into the human. So, of course, for Stargardt's and for other inherited retinal diseases, the method of that, of course, is injecting the gene therapy, injecting the vector into the back of the eye. And once it's in the back of the eye, the reader that we've said here, the person it's being delivered to, is effectively the cells. And what the cells do with their own machinery is that they then have a healthy version of the ABCA4 gene, and then with that healthy version, it can then transcribe and produce healthy versions of the ABCA4 protein. And effectively, the aim, of course, eventually would be to either cure the disease or at least be able to halt or reduce the rate of disease progression at this point as well. So, when we think about how this translates to gene therapy in Stargardt's disease, of course, we start with the disease, which is due to the faulty ABCA4 gene that we were referring to in the previous slide. That faulty gene means that either no ABCA4 protein or a faulty version of the ABCA14 protein is produced. And that leads to that deterioration in the back of the eye in the photoreceptors. And that deterioration, of course, leads to poor vision. And eventually, in some of the more advanced cases, of course, it leads to deterioration of complete deterioration of the central vision for a number of patients as well. And so for the aim for us for the gene therapy is that we're going to inject back into the back of the eye a healthy version of the ABCA4 gene. And once it's injected into the back of the eye will now use that new gene to produce a new functioning version of the ABCA4 protein. And that new ABCA4 protein will help the eye cells function a lot better. And of course, that means that the aim is for patients that are provided the gene therapy, ultimately, the aim is, of course, to provide better vision moving forward. And so, I think, as a community, you'll be aware that there are a number of different companies looking at potential therapies for Stargardt's at the moment, some of which are within gene therapy, similar to SpliceBio, and some are outside of gene therapy as well. And of course, they all have their different potential mechanisms of action in order to provide a treatment. For us, for gene therapy, what we're aiming for is what's called a one and done. And what we mean by that is that we provide the gene therapy into the back of the eye once, and then you should be done for life. There should be no additional therapy that's required there. And of course, the ultimate aim, as I was saying, is to cure the disease. But, to be honest, we're not quite there yet. I think that's going to be 10, 20, maybe a little bit longer years down the line before we're in a position where we can, say with certainty that we can cure the disease. I think, what we're aiming for and what we should aim for at the moment is to halt disease progression. So wherever you are with regards to the progression of the disease, the ultimate aim for us at the moment is to halt that progression any further, once treated. But at the very least, what we want to be able to do is delay that disease progression, or slow down that disease progression as a result of having the therapy. So, when we think about, coming back to, where we are in this and the studies that we have at SpliceBio as well, we have a number of studies that are within our Stargardt's development program. The first one that I think probably people are most aware of is our observational study, or our natural history of disease study, which is called Polaris. And that's now recruiting in the US, the UK, and soon will be recruiting in Europe as well. And participants that are enrolled in the Polaris study will have an opportunity, should they fulfill entry criteria, of transitioning over into our new phase 1/2 first in human study, which we're calling the Astra study as well. And so the Astra study, we've now treated our first patients within the first in human study in the US, and then we'll be looking to obviously continue at a slow rate to enroll more patients into the first in human study moving forward. And and as I said, to reiterate, in order to be considered for the phase 1/2 study, the Astra study, you have to be part of the Polaris, the observational study first. And I will go into the reasons about why that's so important in subsequent slides. And ultimately, the information from the phase 1/2 study, the first in human study at this point, will allow us to, should we continue to see a good safety profile and we start to see the first trends of whether the drug works, whether we get an efficacious effect from the drug. The aim in the short to medium term would be to plan a much larger phase 3 study. And patients from Polaris, again, would transition into that larger phase 3 study. And ultimately, the information that we generate from both the Astra study, the phase 1/2 study, and the phase 3 study would be the totality of the data that we would then provide to the FDA, the MHRA, and the European Medical Agency to submit and hopefully, a filing to get a marketed treatment, should the safety and the efficacy look good. The other new study that we have at this point, which is exclusively running in the UK at Moorfields Eye Hospital, is called the Pulsar study. And that's a new functional visual test that we're looking at. And I actually have a slide on this in a little bit more detail, but just to let you know, so at the moment, in the yellow arrows, we have 3 studies that we're now running within the Stargardt's program: the observational study, the first in human study, and the new functional visual test study called the Pulsar study in the UK. So thinking about the natural history disease study, the observational study, one of the questions we get asked a lot, and it's not just within Stargardt's community, but within the inherited retinal disease community, is, why is being part of an observational study or a natural history disease study so important? So for those of you that don't know what an observational study or a natural history of disease study is, it's a study that where there is no treatment at this point, but it's an opportunity for us to enroll participants that in most likelihood would be the type of candidates that we'd be looking for in the short to medium term to transition over to the gene therapy studies, whether that be the Astra phase 1/2 study or the phase 3 study. And the reason, there's numerous reasons why being in an observational study is important to us. The first thing is, Stargardt's and inherited retinal diseases are complex diseases. And the data that we generate from looking at participants in the real world and how their disease progresses over a period of time, it is very, very powerful for us. One of the reasons why it's so powerful is, in our interactions with the regulators, so the FDA, the MHRA, and the European Medical Agency, if we're able to generate a lot of pre-treatment data, so for example, 8 or 12 months of data within an individual participant before they transition over to gene therapy, and then follow that patient out for another 12 to 24 months, you have this large wealth of information, both pre-treatment and post-treatment on a participant. And of course, if we are seeing a treatment effect, having all of this historical pre-treatment data within the observational study becomes very, very powerful when it comes to us having interactions with the regulators around whether the gene therapy is working. So it is a powerful method for us to increase the likelihood of getting approval of the drug. The other thing that, particularly what Polaris or the observational study is doing at the moment, is it's allowing us to confirm the measurements, the endpoints that we have within our gene therapy studies to ensure we're giving us the greatest chance of success. Whether that be looking at images of the back of the eyes, whether it be reading a visual acuity chart, microperimetry, all of these bits and pieces. It really allows us to pin down and solidify the clinical studies and the endpoints that we have, and again, to give ourselves the greatest chance of approval with the regulators. So, who are we looking for Polaris? So this is a very, very broad criteria that we're looking at, and if you are interested, I'll provide you with information on the clinical trial centres later in the presentation. We're generally looking for patients between the age of 18 and 65 years of age. There is an opportunity for patients under the age of 18, so within 12 to 17 years of age, to enter Polaris, although we do already have a number of these kind of adolescent patients in the study as well. And so, of course, thinking about them transitioning over to gene therapy, we tend to only treat a very, very small number of adolescent patients, and only when the safety profile in the adult patients is sufficient. So we're looking for a very small subset of adolescent patients. And there is an opportunity for us to potentially enroll patients over the age of 65, but it would be a conversation that we would have between SpliceBio, the study doctor, and the patient about whether we feel that they would be a suitable candidate to move forward into Polaris as well. What you would need to do is, if you don't have a genetic report already available from your local clinician, we'd ask you to provide a saliva sample. And this is to confirm that you have the mutations within the ABCA4 gene that would actually make you a suitable candidate for the gene therapy, and actually make sure that you would get a therapeutic effect from having it as well. So the genetic report is a very, very important part of the entry criteria of Polaris. We would need to look at your level of vision, and that's measured by reading letters on a chart as well. We would need to, what we would like to do within the Polaris study is, obviously, as I said, monitor your disease progression over a period of time. And that would be by reviewing pictures of the back of your eyes. So people may be familiar with collecting OCTs, microperimetry, all of those bits and pieces. So there would be an opportunity for us to take those images, during the Polaris visits as well. And of course, they are very high level criteria. The study doctor would also assess other criteria just to make sure that you'd be a suitable candidate to move forward in the Polaris study as well. So switching gears and to give you a little bit more information about our first in human gene therapy study, which is now enrolling in the US. We are looking to recruit around 55 to 70 participants in the US, the UK, and the EU. Participants, as I said, will be selected from the Polaris observational study at this point. And the primary aim of a first-in-human study, such as Astra, is really to establish safety, to make sure that we see a good safety profile at several different dose levels. And of course, the other key thing is, a little bit further down the line, when we've been following the participants that have had the gene therapy out to 12, 24 months and subsequently, is to see whether we're actually starting to see an efficacy signal, i.e., does the therapy work? Are we halting, or are we at least slowing down disease progression compared to what we were seeing in that participant during the Polaris study, when we were just following them prior to having treatment within the gene therapy as well? It's up to 11 visits over a 2-year period that we would follow the participants for. And again, the visits would include visual tests, taking pictures of the back of the eyes, and there would be some questionnaires about, how you think your vision is being affected on day-to-day activities. One important thing, I think, that isn't written in these slides, but it's important just to emphasize is, we know that that we know that it's a big commitment to join these studies and participate regularly in these types of studies. And so what I can say is, SpliceBio have a travel agency that will pay for your hotels, all of your travel, your meals while you're traveling to and from the site, regardless of whether that means getting on an aeroplane in order to get to a center, or for example, whether it just means taxi or bus fare as well. So that is all approved by the ethics committees or the IRBs in the US. And the other piece is that we are able to provide a payment for of £150 or $150 or €150 per visit for attending as well. Because of course we know that in a lot of cases, either whether you're a parent, a carer, or you're a potential participant, it's taking time off work as well. And so we know that that amount of money isn't going to necessarily replace the loss of pay or holiday that you have to take, but it is a small token that we were able to get accepted by the ethics committees and the IRBs to move forward and make sure that we can make the visits as smooth as possible for you as well. So the final piece was the new study, which is exclusively in the UK and at Moorfields. So this is really going to be thinking about these patients that are based in the UK at this point. So SpliceBio and Moorfields, we're running a new study called Pulsar, and it's to assess a new functional vision test. The reason why this is important is because, increasingly, the regulators, when you provide them the data at the end of phase 3, they want to know that in addition to maybe seeing the photos in the back of your eyes that you can see a disease progression, it's also really important to the regulators that the participants or patients actually are seeing some kind of actual functional benefit, some kind of subjective benefit of the treatment as well. And so one of the things we're developing is potentially this test that we would integrate into the gene therapy studies. So this test is very, very important for us to generate data and to see whether it's something that potentially we could use further down the line to increase our chances of success in the gene therapy studies. And so what it involves is basically selecting objects at different light levels when prompted by a computer. And the aim is to reflect everyday activities such as picking items off a shelf in a shop or picking up toys or bits and pieces like that. So something that you would do on an everyday basis. It's actively recruiting now. We're looking for participants at Moorfields to attend 2 or 3 visits over a 28-day period. You need to have a genetic diagnosis of Stargardt, so you would need to have a historical genetic report available. And if you are interested, then you can contact us at clinicaltrials@splice.bio for more information. And just to let you know, so again, in addition to what I've just described in the Polaris study and the Astra study, the first in human study, your expenses for traveling to the center and inconvenience fee will be paid. So we will pay up to £450 for the 3 visits that occur within the 28 days as an inconvenience fee as well. So this is one that we're actively looking for participants. So again, as I say, just to emphasize, if you are interested in helping us with this functional visual test in Moorfields Eye Hospital, please contact us at clinicaltrials@splice.bio. And so generally, when we're thinking about, if you are interested in joining our clinical trials, there's different ways that you can get involved. So the first one is through our website, www.splice.bio. You can email our team at the same email address as I've just described, so clinicaltrials@splice.bio. And what the clinical trials team will do is, if you provide us your consent that we can use your data, and we've got your location and email address, we can direct you to your nearest clinical trial center, either in the US, in the UK, or will be in Europe as well, to sit and for the clinical trial center to reach out and assess a very broad level whether we think that you would be a suitable candidate to move forward in any of our studies as well. And this is specifically just at the bottom here for the folks from the UK. We are enrolling at Moorfields, Oxford Eye Hospital, and there are other UK sites opening in the north of England, that we're assessing probably in the next 3 to 6 months as well. I would say in addition to that, for our participants on the call outside of the UK, we are in conversations with the European Medical Agency at this point around the opportunity of opening up centres within Europe as well. So that's something that, there will be an opportunity to enroll into Polaris. Which countries we'll be able to go to at this point is still a little bit under discussion, but we are actively looking at expanding the program into Europe as well and giving an opportunity for participants to join outside of the UK and the US at this point. And that was it. That was the presentation. So we've done about 35 minutes. So I think probably got about 10 minutes for questions, something like that.
So if anyone would like to ask a question, they can either